Well, thanks everybody for coming, and sorry that I'm not there in person. I'd signed up to organize the meeting with Christian and Kavita back a few years ago, and since then had a toddler and realized it was a bridge too far to either bring him or leave for that long. But anyway, what I want to talk to you about today is some work that we've been doing in my lab on the genetic architecture of repeated adaptation and looking at local adaptation versus global adaptation, mainly in plants. And I guess provocatively trying to title my talk here, surprisingly consistent with theoretical predictions. I'll talk about a lot more than that, but hopefully we'll see what's there. So I wanted to start off by thinking about what repeated evolution is from maybe most familiar forms, where commonly we think about things like convergent evolution in wings being a really celebrated example of repeated adaptation. Oh, and sorry, just before I get going on this, hopefully I can hear people. I know it's been the talks I've seen so far, there's been a nice kind of banter back and forth. I hope that people can interrupt me if you have questions along the way. I should have lots of time to cover everything. So please, I don't know how well it will work with Zoom, but please do try that out.
Yeah, we have
Yeah,
mics.
so yeah, great. So yeah, please do interrupt and ask questions at any point, because it's nice to have this be fairly informal. Anyway, so with convergent evolution, I think what's fascinating about this is not so much thinking about why the same thing evolves over and over again, but why we don't see other kinds of things evolving. And there's 400,000 species of beetles. We might kind of think, why is it the case that none of them have evolved wheels? And it's a bit of a silly example because there's all sorts of biological constraints that will prevent articulation of a joint in the way that you need for a wheel. But I think that this gives you the idea of what I think is almost the most interesting part of convergent evolution or repeated evolution is understanding what's not happening, why it is that we're seeing one thing over and over again and not some other forms. What is it about those other forms that are just absent or just not possible or low fitness? And so to bring this into the genetic context, this is a really celebrated example that we see in textbooks and review articles all over the place. MC1R gene driving color variation in vertebrates. So local adaptation driving differences in populations that inhabit different areas and seeing that many different vertebrates use MC1R to adapt to these varying color background environments. And of course, you might think, why is it that the case that analogous to the kind of beetle in the wheels, why is it the case that we see MC1R and not all sorts of other genes that can affect coloration? And we know from this is a nice review done by Hopi Hoekstra looking at all the different genes that can affect coloration in mice. Just a huge list of them. I'm sure there's been probably more since this list was compiled that have been discovered. And MC1R is, of course, just one of these many genes. Of course, many of them will drive color pattern in ways that are not great. And so a lot of the discussion around why MC1R is contributing over and over again is that there's limited negative pleiotropy, not so many deleterious consequences when you have mutations in particular parts of MC1R. There appear to be many different ways that you can mutate to cause loss of function of the MC1R receptor. And so you can see here that the mutation sites are shown in colored circles here on this cartoon version of MC1R. You can see lots of different places where mutations have been identified that drive this color polymorphism. And so I think hopefully this sets the stage for what I think are the really interesting kind of big talks or big questions that my research and many other people's tries to get at in this realm is understanding how repeatable adaptation is for more complex traits than color variation. And then why repeatability might vary among genes, traits, or species. And I think these kind of aspirational questions are certainly not ones that I can answer in this lecture, but I think particularly this is what I'd like to see us working towards within this kind of sphere of research. And so what I'm going to talk about today, I'll start with a review of some population genetic theory on repeated adaptation, and then look at examples that we've worked on in my lab in sunflowers, then in 25 seed plant species, looking at local adaptation to climate, and then looking at global adaptation, so selective sweeps in the same set of seed plants that we look at local adaptation in. So let's start off with a review of the theory and kind of getting all on the same page. Some of this, I'm sure, will have been talked about in the lectures that happened past midnight for me that I missed. So hopefully it's not too much overlap with what you've already been exposed to. But what I thought I'd start with was just throwing up some straw man examples from various different schools of thought that you might naively kind of extrapolate from. So why do we observe any repeatability at all? And based on, say, quantitative genetics or the infinitesimal model, universal pleiotropy, these kinds of ideas assume that many loci with interchangeable small effects are kind of the basis of trait variation. And if that's the case, we might naively expect low repeatability. If you take a kind of a population genetics lens to evolution, you model each allele experiencing selection, and it's only by adding in really complicated models with epistasis that you can kind of generate other kinds of outcomes. So if every single allele experiences selection, we should expect to see higher repeatability, I think, kind of naively. But of course, this would be limited by waiting time for mutations to come around. If you ask a molecular biologist, they might say something like each gene has a specific function, and maybe there's not a lot of redundancy in how genes do things, and so they might expect high repeatability. Whereas if you ask somebody that works on GWAS, where they identify hundreds to thousands of loci underlying many traits, they may kind of imagine that this suggests low repeatability. So I don't know to what extent somebody that would call themselves one of these particular biologists would agree with me on this, but I hope that these caricatures show you that I think expectations for how much repeatability we should see really depend on your background and your lens, and I think there's expectations all over the map. And so what I'd like to kind of think a little bit more about is how can we bring these different lenses into focus together, and what that might tell us about what our predictions should look like, and then how do we compare our predictions with what we see in nature. And so to do this, I'll present a caricature model that a lot of simulation approaches have taken, which is to look at what I've been calling genotypic redundancy, which is the amount of the difference between, say, the number of redundancy is kind of this thing where you're kind of saying if there's an optimal trait, in this case signified by this genotype here, your genotype needs 5 mutations to get this nice red tree here. You would have a high redundant situation if your genome had 500 loci that affect redness. So there's many, many more loci that drive trait variation than there are mutations that you need to get to a phenotype. And so you could say that's a case with high redundancy. If there was a different situation where you had 5 mutations to yield an optimum, but you only had 5 loci that could mutate to give you redness, then you would be in a no redundancy situation. And anything between 5 and 500 then is some intermediate amount of redundancy in this example. And so here there's a quantitative way of explaining how much redundancy there is in a trait based on the difference between the number of mutations you need to get to some optimum.
Sam?
Yes.
Oh, just a quick question. So this is all assuming additivity at this point?
Yeah, sure. This is just a cartoon model. I mean, I'm not going to use this in any quantitative sense right now. I'm just kind of saying this idea, there's something like this where we think that, if you need a certain number of mutations to get somewhere, and there's many, many different genes that could furnish those mutations, then it's a highly redundant case. Certainly, if there's epistasis, that's going to change the exact nature of the mapping. So I'm not going to attempt to make a really concrete definition of redundancy, but
okay, thanks.
More just as a qualitative idea that there's all sorts of different models you could formulate that would make a precise version of this, but just that this is some quality you could look at in any model that you could actually tune, and allows you to go from kind of a more population genetic model to a more kind of quantitative genetic model. And hopefully that becomes clear in a moment. So redundancy really impacts how adaptation works. If the only thing you change is the amount of redundancy in a trait, here these are two simulations where we've got individual-based simulations with either 50 loci on a chromosome or 500 loci on a chromosome, and we're looking at the last 5,000 generations of a 50,000 generation run of a simulation. And here we're looking at local adaptation, so two populations that are adapted to different environments with migration between them. And the blue or the red colors are cases where a locus is contributing a lot to the divergence in allele frequency between those two populations. And you can see that the low redundancy case is highly stable, whereas the high redundancy case is got this transient architecture. Both of them have divergence in the phenotype, but there's a difference in the underlying architecture. And that's only caused by the only parameter that was changed here was the number of loci. This has also been seen in models of global adaptation, so where you've got the same environmental optimum in different populations or in just a single population. A single population has experienced a change in environment. A very nice model by Ilse Hollinger, who was working with Joachim Hermansen, showed that if you scale the amount of redundancy in a model where you have adaptation to this change in environment, you can either go through a regime where you have, if you change in allele frequency and each one of these points is a simulation replicate, you could have cases where there's just a few large allele frequency sweeps. So cases where there's many replicates have very, very large changes in allele frequency. Or you can have, with high redundancy, cases where there's many, many shifts in allele frequency. And so a highly polygenic response with changes in allele frequency at lots of loci. And so again, the only thing changed here is the amount of redundancy in these simulations. And so what I think is really neat is that we can see that, just changing this parameter in polygenic adaptation models scales really dramatically the kind of behavior that we see in how adaptation works. And that's for local adaptation, where there's heterogeneous environments with migration, or for a global adaptation where one population or several populations linked by migration are all experiencing the same new optimum. Now, our models for studying adaptation kind of make implicit assumptions about redundancy. If we go to the most basic forms of these models, population genetic models, where we look at P prime, or P as a function of, should have had a P prime here on the left, but change in allele frequency at a single locus is implicitly assuming that there's no redundancy. Redundancy would come in if you modeled epistasis between the loci. So classic simple population genetic models without epistasis are kind of like tuning redundancy down to having absolutely none. Whereas the quantitative genetic models, where you have some kind of stabilizing selection, are implicitly making an assumption that there's lots of redundancy, that there's lots of different ways of getting to your optimum phenotype. And so I think what's nice about these kind of simulation-based approaches to polygenic adaptation is that it allows you to explicitly tune this parameter and kind of alternate between behavior that's more population genetic-like or behavior that's more quantitative genetic-like to kind of characterize those different worlds. Is that all making sense so far? Any questions? So what I'd like to talk about now is just yes, please.
It's on. For your example with the temporal change in allele frequencies over time, I guess about 2 slides back, Sam. Maybe one more. Yeah, so in the case of the left, I guess you're talking about stabilizing selection. So what happens if the 6 loci there don't sum up to the optimum?
Well, they won't sum up to the optimum because the optimum amount of divergence under this amount of migration selection is kind of sub-fold divergence. But yeah, in this case, if you allow allele frequency, the allele effect sizes to also evolve, I think in these simulations I held the allele effect sizes to be constant. They were just a diallelic or a triallelic level. But if you allow the allele effect sizes themselves to also evolve, weirder behavior starts to happen as well, which I could talk about at length. But basically, in these models, if you allow stacking of mutation effects over long periods of time, you'll get what I've called concentrated architecture, where a single QTL ends up contributing all the divergence between populations, because that's a very perfectly linked arrangement that has no recombination. So I mean, if I can, in these examples, I constrained the allele effect size to not evolve so that it would preclude that behavior. I don't know that answered your question though, Mike.
Yeah, I'm not sure, but next slide.
Okay. So I think what I hope you can see is that the repeatability also of these kinds of scenarios is going to differ a lot, right? And so we can start to make predictions about repeatability based on the amount of redundancy. And clearly, I think it's quite intuitive that if you have very, very high redundancy, you're going to have very, very low repeatability. But I think it's also interesting to think if you have repeatability, where is that coming in? Where is the low redundancy coming in? And it could be that low redundancy in your genotype to phenotype to fitness map is coming in because there's just not many adaptive phenotypes that you can generate. You know, there's just not a lot of ways you can actually mutate and make a change in the trait that needs to evolve. That's one kind of extreme of a worldview. Or you could be in the other extreme of a worldview where there's lots of phenotypes that are so-called adaptive, but then not all genotypes have high fitness. And if that seems a bit esoteric, this is the kind of thing I'm talking about here, where, you know, if you've got a very modular trait architecture where, you know, there's very, very limited pleiotropy and very, very circumscribed effects of genes, then there's just not very many ways that genes can affect your trait of interest. Whereas it may be the case if you've got a highly integrated trait architecture, there may be many, many genes that can mutate to cause a variation in your phenotype of interest, but that concerns like population genetic concerns like the drift barrier, S versus NE, or migration selection balance, S versus M, or pleiotropy, where there's negative consequences of some mutations but not others on other traits that haven't been measured. All these kinds of factors might constrain why we see repeatability and favor certain mutations that are, say, more favorable for these various different properties. And so I think these are kind of two very different ideas about why repeatability might happen as a function of limited redundancy in that genotype to phenotype to fitness map. So I think this is, again, a very nebulous kind of qualitative ideas that I think we could formalize quantitatively in all sorts of different ways. But I think it's really interesting to try to understand which of these worlds do we live in when we inhabit different species, or, you know, when we're looking at different species, different traits, what kinds of things matter. And I think I'd really like to understand this ultimately through my research, but I think we're a long way off. So I think understanding how much redundancy is out there is a really important thing. It, as I've shown really briefly here, it really affects how evolution and how adaptation behaves, whether you have high or low redundancy. So it's something we want to know about. And what I'm suggesting is that we can understand redundancy by studying repeatability. Because if we see high repeatability, it must be the case that there's been low redundancy at some point, and vice versa. And so I think what I'll mostly be talking about today is just how different factors affect repeatability and what we can learn about redundancy through studying repeatability. So I want to just review a bit of theory, looking at some other factors that are important. And I imagine this has probably been talked about a bit in the lectures I've missed. So I may be reviewing stuff here. But this is a nice review by Conlon and Hodgins looking at the Fisher-Kimura-Orr model of adaptation, where you know, there's some change in the current phenotype and the optimal phenotype. We're evolving towards some optimum, and mutations can take on a particular size. And Fisher showed that the probability of a mutation, randomly chosen mutation, occurring and improving the trait is kind of a decreasing function of the mutation's effect size. Kimura added in fixation probability to look at the probability of the first mutation in an adaptive walk fixing, and showed that intermediate size mutations would be most favored. And then, or in some classic papers, showed that the distribution of allele effect size fixed over an adapted walk, so many mutations, as you traverse this space towards the optimum phenotype, would take on a form that's approximately exponential. And intriguingly, he found this was robust to different assumptions you might make about the form of distribution of mutation effect sizes going into the adaptive process. And so I think this is a really neat result that really showed us that evolution should involve both large effect mutations early in the adaptive walk process, and then smaller effect mutations later on as we approach this optimum. And this Fisher OR model has kind of been talked about a lot in terms of things like the cost of complexity, and I'm here talking mostly about why we understand pleiotropy mattering. And in this model, pleiotropy is just the number of different traits that you have that you're looking at that are required to move from your ancestral phenotype to the optimum. And if this cost of complexity idea came from some work by Alan Orr and others, I think, looking that basically if you have more traits that are possibly contributing to adaptation, then there's a greater chance that mutations will be going in a wrong direction, and overall this will result in a slower rate of adaptation due to mutations disrupting traits in a negative way. Some really nice work has been done by Luis Miguel, who I think is in the audience, although I can't see in the tiny window. Hi, Luis. And Martin and Lenormand, looking at repeatability being lower with higher pleiotropy. And so they found that if we looked at the repeatability of adaptation among bouts of evolution, if you had cases with higher pleiotropy, so here this is the number of trait dimensions on the x-axis, this index of repeated adaptation that they were calculating would be lower as you got to a larger and larger number of trait dimensions. They also allowed the amount of pleiotropy of different genes to vary, and showed that genes that are lower in their amount of pleiotropy are more likely to contribute to adaptation, and then that can drive higher amounts of repeatability. And so this building on this kind of Fisher-Orr model, there's working towards this understanding of how pleiotropy interacts with repeatability, and I think quite intuitively thinking that lower pleiotropy will result in kind of higher genes with lower pleiotropy will have a higher likelihood of contributing to adaptation repeatedly. So there's that kind of simple prediction. What I wanted to do is contrast some of these models with local adaptation models, and just review the really basic form of these models as the continent island model, where there's, say, a huge continent population in red here, sending migrants to a small island population in blue. So the blue background is indicating the environment, and the genotypes here are these little trees that are either a locally adapted allele in blue, or a maladapted allele being immigrating to the island from the mainland, unidirectional migration. And even back in 1930, when Haldane first formulated this, he showed that if you have the migration rate M being greater than the strength of selection S, then you're going to have swamping of local adaptation on the island. And so a high rate of red, when M of the rate of red alleles moving into the island is greater, then the blue allele will be lost. And we can kind of take this to predict that large alleles should be more likely to contribute to local adaptation than in the absence of migration. And so these, just drawing here, alleles that could contribute to adaptation and their effect size, these ones that are small effect, are not going to be contributing to adaptation stably, and so we should see this enrichment for large effect alleles. You can also look at the effect of linkage among alleles, And if you cluster different alleles close together on a chromosome, then recombination is not breaking them up, and they can act like a single large effect locus, and they can kind of get over that hump of this migration swamping boundary due to M. Some really nice work, of course, by Nick Barton and Mark Kirkpatrick and others looking at how inversions can do the same kind of thing here, so contributing to local adaptation. So inversions can suppress recombination and allow the generation and maintenance of LD within their boundaries. And so there's this broad prediction that when you've got local adaptation, we should see inversions capturing locally adapted loci as well. So I just wanted to walk through some different examples of genetic architectures that we can see under local adaptation when we have very, very low migration on the left versus kind of intermediate to high migration in the middle, and then high but still subcritical migration where there's still divergence in the mean phenotype here, but a total loss of stable polymorphism. And what you can see is that when you go to these intermediate migration rates, the benefit for large effect mutations causes there to also be a benefit for clustering. And so over very, very long periods of time here, this is in hundreds of thousands of generations. So long periods of time, we see a kind of clustered architecture evolving under migration selection balance that doesn't happen at very, very low migration rates. And so you can kind of think about this as being like global adaptation. We've got adaptation happening basically without migration. You got really no effect of that on clustering, whereas when migration is happening, larger effect alleles are favored and clustering is favored if it's possible.
Sam, what's the horizontal axis?
The horizontal axis, sorry, I should have written, is time. So here, the same kind of framework, we're looking at position along a chromosome here, and then the horizontal axis is time in hundreds of thousands of generators. So this is like 100,000 generations, 200,000 generations, 250,000 generations. And the points are showing, if there's a white point on this map, it's showing that that's a locus of the genome that is not contributing to divergence between populations, whereas the colored points are contributing either, if it's reddish and yellowish, it's going in one direction versus bluish, it's going in the other direction. And so here you're seeing early on in the simulation, there's lots of, these ones are very, they maintain almost the same amount of phenotypic divergence between populations from the beginning of the simulations to the end. But then the genetic architecture underlying that gradually evolves and becomes this highly clustered form. And this occurs as alleles that are tightly linked fortuitously tightly linked invade and replace alleles that are loosely linked. And so there's a competition among genetic architectures in this case here. And that competition is not happening when there's a lack of migration, because there's no benefit for yes.
Yes, just to make sure the phenotypic differentiation remains similar across this regime here, or?
So these ones here would be the most differentiated. These ones here would be kind of intermediate, and these ones would have lower divergence, but they would still have a stable amount of divergence at equilibrium.
So are there some parameter values where you could maintain the same phenotypic differentiation, but through very different genetic architectures, or?
Yeah, you can, as you ramp up the amount of genetic variance, so as you increase the redundancy under any of these cases, you would increase the amount of divergence in mean phenotypes, but then you also increase the amount of load that's segregating in the form of mutations present.
Oh, yes, sure. Okay.
Yeah, so that is one thing that can happen. And actually, really intriguingly, these concentrated architectures will be swamped out by mutation. As you get higher and higher per trait mutation rates, at some point, you can actually just evolve enough at your genetic background so that the large effect alleles just get lost, and you actually transition into this transient architecture. Any other questions about this slide before I go on? Okay, so just showing here two different ways of looking at, this is on the left-hand side, a nice paper by Kurt Griswold, looking at directional selection for 1,000 generations and showing the case with no migration is in the dashed lines here. And this is the allele effect sizes that are contributing to divergence, to local adaptation and divergence. Whereas the case when there is migration, you can see that that distribution of alleles is shifted towards larger and larger effect loci over time. This is the paper by myself and Mike Whitlock looking at the same thing, but over much deeper time, so looking at 2 million generations. And you can see the allele effect sizes that are contributing to divergence between two locally adapted populations. Starts off being dominated by small effect alleles, and over time, a QTL evolves that explains most of the divergence between populations. So when you have this local adaptation dynamic where there's divergent selection pulling populations apart and migration homogenizing the differences between them, that generates this pressure for a gradual concentration of the architecture towards this really highly clustered thing. And what I think is interesting about this is that we could extrapolate to think about how this might matter in the context of pleiotropy, because some empirical data has shown that genes with higher pleiotropy tend to also have larger effect sizes overall. And so here you can see some data from Wang et al. Study looking at the degree of pleiotropy, so the number of traits that are affected by mutations, and then the allele effect size. And you can see that mutations or genes that were more highly pleiotropic were having larger allele effect sizes. And we may just naively think, with this empirical idea, perhaps migration selection balance should favor higher pleiotropy than global adaptation. Recalling back to when we were talking about global adaptation, just the Fisher-Orr model, we expect that because of the fine-tuning problem there and having lots of traits be affected, a pleiotropic gene will really have trouble fine-tuning adaptation. And so we have this prediction that low pleiotropy should be favored in terms of repeatability and adaptation with global adaptation. Perhaps because of this benefit for large effect alleles, we should see higher pleiotropy being favored under migration selection. To look at this a little more formally, I did some work with Paul Batley and Kay Hodgins at Monash, and we looked at a two-patch model with mutations affecting multiple trait dimensions. And to keep it simple, we just looked at 5 QTL, 1 focal locus, and 4 background loci. And you can kind of think about this as being similar to the core versus peripheral classification of genes in the omnigenic model. So we wanted to have 1 focal locus that was, say, a really big effect locus, and then other loci that were just maybe small effect loci, and see how that would work when we had either 2 configurations of environment. So either what we called beneficial pleiotropy, when the direction of, if we kind of made an underlying mutational correlation to our 2 traits, so that mutations tend to either increase trait 1 and trait 2 or decrease trait 1 and trait 2, we would hold that underlying mutational correlation constant, and then make the environments between the 2 patches that these populations were inhabiting either aligned with the same axis as that mutational correlation, or off axis, where they're divergent in one trait, but non-divergent in the other trait. And so we were curious in seeing how do pleiotropy and migration affect repeatability in this model of local adaptation. And I won't go too far into the weeds with this paper because it gets kind of complicated, but all I want to show you is that when we have a zero migration case on the left, so that's kind of like a global adaptation case where there's no migration pressure, versus migration on the right where there's local adaptation, we see more repeatability under the migration case, and the proportion of adaptation that's driven by a locus of large effect is higher for any scenario we look at under the local adaptation case. And that's the case, either beneficial pleiotropy, so that's this case on the left where there's alignment between the mutational correlation and the divergence in environmental optima between populations, or deleterious pleiotropy where there's a mismatch between mutational correlation and environment. In both cases, we see migration causing there to be higher repeatability, and this large effect locus driving more of the difference between populations. So even when there's pleiotropy in the wrong direction, we still see this effect happening more under local adaptation. And so this kind of, I think this supports this idea that we might see increased repeatability and importance of large effect loci and pleiotropy with migration. So I'm going to wrap up the theory part that I'm talking about here with just summarizing some of these predictions from this, that local adaptation should involve alleles with larger effect sizes, and that repeatability will be driven by genes with higher pleiotropy when we're looking at local adaptation as compared with global adaptation. So that's the kind of main prediction that I'll be looking to test later on in this talk. And then I'll also be looking in the sunflowers part at inversions. And so, just this idea that inversions are important for local adaptation and kind of understanding repeatability among many species. Again, just reminding us, I don't think we have any really clear predictions about how much repeatability we should see, because what we predict about repeatability differs depending on which model we're using. And so I think this is really a case where theory doesn't have a lot to tell us about how much repeatability we should see. And it's more the case that empirical data will tell us how we should be making our models. Are there any questions before I go on to empirical stuff that I could clarify any of that theory preamble with? I'll go ahead then. So I'm just going to talk first about sunflowers. This is older work that's now been published.
Hello? Sam?
Oh, yes, question.
Sorry. This prediction that under local adaptation, genes with higher pleiotropy could play a more important role. This is based on this stronger, this positive correlation between pleiotropy and effect size, right?
I think that's some empirical grounds for this. Yeah,
Yes.
I think that there's a purely empirical justification, and then I think also some very, very not strong effects in theory.
Effect size on a particular trait or total selective effect? What is this effect size?
This is the total selective effect, I think.
But if you sort of have local adaptation that involves, one or two gradients in a small number of traits relative to the total number of traits, then I imagine that it's, which might be the case. And then the relevant effect size is, I guess, not the total effect size, but, the effect size of, say, if gradients involve one trait out of many. So, I mean, it isn't obvious to me what really the prediction is, for this.
I think that's valid, and certainly, I'm making a few leaps here. I don't. Yeah, I agree with you on that. I think it's the case that when we have local adaptation, there's often lots and lots of traits that tend to differ, and maybe over long periods of time, the actual G matrices or the underlying trait correlations evolve a little bit in response to that. But that's a more complicated question than I've even approached at all here. The theory part here, which I didn't want to spend a whole lot of time going into this paper, but what we were showing was just that we're still, even in this model here, this case on the right, the deleterious pleiotropy case, is kind of like what you're saying, where there's one trait is diverging between populations. That's the Z1 trait, where the populations have an optimum at -10 versus 0, and then Z2 trait, they have the same optimum. That would be kind of like your example of, if a trait is pleiotropic, only one of the trait dimensions is actually experiencing divergent selection and the other dimensions are not experiencing divergent selection. So this deleterious pleiotropy model is a bit like that idea you had, I think, right? Is that what you were getting?
So,
and then...
Sorry, did you finish, or I had a...
yeah, go ahead. Yeah.
Yeah, sorry. So this is maybe a question about the structure of local adaptation, because I think you've been talking so far about a single gradient, which might involve a lot of traits, but you can always kind of rotate things so that you're defining the trait that matters as the one that changes along the gradient. But you can imagine situations with a kind of mosaic habitat where lots of things are changing with multiple overlapping gradients and so forth, maybe more easily in a sort of island model than in a continuous population, but maybe in both. I'm just wondering how you think about the complexity of the challenges a population faces in this relation.
Yeah, I think that's really the next frontier for theory modeling is exactly that. I think this is how populations respond when there's cases like underlying trait correlations that may affect many of these the evolution of many of these traits, but then the environments are varying in a mosaic way that's not parallel at all with that dimension. And I think that the model that we were working on here is very, very simplified. I think there's a lot of scope for looking at that. What I was surprised in this model, and I don't have the slide to show it, but the actual mutational correlations that we were simulating didn't present particularly strong constraints to evolution. It would just take longer for a suitable mutation to occur. And so we'd kind of have stacking of mutations that would kind of bring you a little bit in the right direction or not perturb you too badly in the wrong direction. And I'm not sure that is the way nature works, but it was to me, surprising fact of the bivariate normal distribution or multivariate normal distribution is that it's not actually a super, super strong constraint. And so I think in some cases, to the extent that that's a realistic model of evolution, that's exactly what, say, Orr was saying with the cost of complexity idea, is just that the rate of adaptation just becomes a lot slower because you've got to wait longer for a suitable mutation to come along that is falling off the usual axis of your correlations.
Okay,
but yeah, I think there's a lot of novelty to be done in that area. I think it's a really neat idea, Nick. I think that was Nick, right? So we can come back to the theory. Let's move on to the empirical stuff, and then hopefully we can have a bit of a discussion at the end about where theory has to go with this. So the sunflower work I did was in collaboration with Lauren Riesberg's lab and some postdocs working with me, Shageg Sudi and Mojtaba Jahani. We were looking mainly at two subspecies of Pediolaris, and Pediolaris falix, and Helianthus annuus and Helianthus argyphilus. So this yellow taxa, the green taxa, and then these two blue taxa here. We were looking at how these four sunflowers or species adapt to abiotic stress, and looking at samples collected from across mostly United States, a few populations into Canada, and a few into around the border of Mexico. And the big questions we wanted to look at was whether the same regions of the genome of these sunflowers were contributing to local adaptation in multiple species, and what was the importance of inversions. So there was some work done by Lauren's lab showing how the large number of inversions in these species, and we wanted to really specifically study the impact of these inversions on repeatable adaptation. And so asking whether regions that harbor inversions in one species also tend to contribute to local adaptation in another species. And intriguingly, there's many inversions identified in each species of these sunflowers, but each inversion tends to be particular to one species. So inversions that are present in the Pediolaris species are not found in Argyphilus, and inversions in Helianthus are not found in Pediolaris, etc. And so what we did to do this was we looked at whole genome shotgun sequencing of about 1,500 individuals across all three species, and then looked at GWAS and genotype environment associations. So GWAS being associations with phenotypes. We had 71 phenotypic traits measured not completely in all of the species. And then we had environments, 15 soil variables, and then 26 climatic/geographic variables. and to look at environmental associations. And so we want to look at associations between allele frequency and either phenotype or environment, and then ask if the same genomic regions in different species were driving adaptation in these different variables. Now, a huge problem that we have to contend with in any observational study like this is that drift.
Sorry, Sam, just a quick question. What's the LD
Sure.
block structure size of these species?
It's really variable. It depends a lot. They probably, on average, decays within under 10 kb, but there's large regions of extended LD around any of the things that we find to be having strong signatures of selection. And of course, large areas of LD within any of these inversions. So it's really highly heterogeneous. I'm not sure of exactly the rate of LD decay on average genome-wide, but it would be under 10 kb. They're highly outbrowed and lots of outcrossing. Does that answer what you were looking at? So one problem that we really have to contend with a lot whenever we do this and think about this is that this is not sunflowers. This is other work I've done in conifers, but just to give you an example of what things look like, this is the MDS decomposition of genetic variation genome-wide, and you can see a blue kind of structure grading into a reddish structure at the bottom. So lots of demographic axis of divergence between these populations due to genetic drift. In this case, these are two species of spruce that hybridize, and so they form this huge hybrid zone through Western Canada. And that main axis of population structure along the hybrid zone corresponds completely with an axis of adaptation where in Northern Canada, they have to deal with very, very cold temperatures. And this is a cold hardiness phenotype measured in a common garden. You can see the amount of cold injury is very, very low in the north and very, very high in the south. And so there's this big problem when we're trying to understand local adaptation in nature is that drift is kind of completely confounding the structure of adaptation. And so the demographic axis is largely oriented along the same axis as environment. And so it's really hard to disentangle the signature of drift from the signature of natural selection. Now, people have come up with all sorts of ways of trying to correct for population structure, where you add some kind of covariate to a model, or you do some kind of something like that to try to adjust for what you think the background amount of population structure due to demography, the effect of drift is on your patterns in the genome, and then find signatures of selection that stick out from that. But I think what I've realized after looking at this for a long time is it's really hard to do that without incurring the opposite kind of problem. If you don't correct for population structure, you get tons of false positives. All sorts of things that are neutrally evolving look exactly like things that are adaptive. Whereas if you do correct for population structure, you tend to reduce the signal of true positives. And so you end up getting negatives where you would like to see actual signatures of adaptation. So what we've taken is two different approaches to detecting repeated adaptation that just avoid the problem of structure correction at all. So we start by doing genome-wide contrasts across pairs of species. And so we're kind of assuming that these, when we're looking at pairs of species, that they're evolving approximately independently. And then if we see things happening in one species that are also happening in the other, that's unlikely to be due to drift. We also can look at gene by gene contrasts. So we look at many species and just say, is this one gene doing anything interesting in several species? And both of these two approaches are built on this assumption that if drift drives a strong association in one species, there's a random chance of it also driving a strong association in the other species. And so to the extent that assumption is true, that allows us to avoid this problem of population structure correction, because we're not trying to identify with confidence what's happening in any one species. We're using multiple species to correct for that issue. So I'll go through the methods a bit in detail just to get an idea of, so we all understand what we're doing specifically with this. So we start off with, for example, a genotype environment association where we calculate allele frequency environment correlations. So here there's number of frost-free days of a given population and the allele frequency in that population. And you can see there's a strong negative correlation between those two quantities. We can affix a p-value to that based on whatever statistic we're using. And then rank our SNPs based on their negative log 10 p-values and consider, say, the top 1% of our SNPs by statistical significance being outliers. We can then ask, are there particular regions of the genome, maybe candidate genes that have large numbers of outliers relative to what you would expect by chance under a binomial distribution? So here's the number of SNPs that we're seeing in a given gene, and then the number of outliers per gene. And we're just saying that any of these genes that are falling very, very high on this outliers per gene relative to SNPs per gene are putative candidate genes in one species. They're things we think might be interesting. We think that selection should be driving a lot of linkage disequilibrium. So very, very strong selection should cause a lot of correlated responses at other SNPs in the same gene. And that should lead to this kind of gene-wide effect with lots and lots of outliers in a given gene. And then in this pairwise analysis, we go to another species and say, if we look at the orthologs of the gene from one species, how many of the orthologs are falling into the tail of a null distribution in the other species? So we'll say, we take this gene here in one species and we say, for the ortholog in the other species, is it falling into the 5% tail of a null distribution of how strongly associated it is?
Can you hear me? Yeah, now.
I can, yes, yeah.
How do you deal with the fact that you have genes in inversion without inversions? Your panel 3, basically, you would expect genes in inversion would also, even under neutrality, would have more top candidate genes than genes outside inversions. How do you deal with this?
That's a great question that will hopefully become clear in a few slides. And if it doesn't, please ask me again. So what to put it briefly, what we do is look at the distribution of these things inside and outside of inversions, but that'll become a little bit more clear. So what I'm going to call these things for now is windows of repeated association, so WRAs. And so anytime we've got a gene that looks interesting in one species and then also falls into the tail of a distribution in the other species, we'll call that a window of repeated adaptation or association. So WRA. So that's one of the two ways that we're looking at this is pairwise contrasts, where we look at strong evidence in one species and then ask if it's shifted in the distribution in the other species. We also used a gene-by-gene approach, so looking simultaneously across all the different species of sunflowers, using genome scans to identify genes that were repeatedly associated with adaptation. So here we rank genes within each species based on the evidence for association with environment. And then we call orthology across the species, so just to match up genes from one species with another gene, or with a gene from another species. And then when we've ranked the genes within each species, we just say, are they looking particularly weird relative to what you would expect from a beta distribution? And so here, this is an example of if you're looking at 15 species and you rank those species across a given, you've got an ortho group that's got a member of a gene, a gene family has got a member in each species. This is the species with the top ranked gene, the species with the next top ranked gene. A beta distribution will describe how strong those ranks should be. And if we're seeing, this is the box plots are showing us the random expectation. If we see this observation where the top 5 species all have very, very low ranked genes, very, very strong association in that species, then this would have a significant p-value. And so we can calculate the p-value for a given gene being strongly associated across several species using this beta distribution to get a nice p-value out of it.
So Sam, a question on the beta. It's a 2-parameter distribution. What are you using to fit the null?
So this is for with a uniform distribution. I forget which parameter it is, but one of the 2 parameters is fixed. Yeah. Good question. So when we called this method PICMIN, Tom Booker was the lead on it, and he chose the name. I forget why. I think it's after a video game. But using PICMIN to test for significance in these sunflowers. So this is the kind of simplest result, is that we found 71 genomic regions across the multiple species at a false discovery rate of 10% that were significant for at least one variable of phenotype, with the strongest environment having 10 regions at an FDR of 10%, or 32 regions at FDR of 2%. The strongest phenotype was days to flower, which had 4 regions or 18 regions, depending on your significance. So we could find regions of the genome in these sunflowers that were repeatedly adapted. Days to flower, we know flowering time is this gene that is strongly associated with responding to photoperiod. And so that was nice to see that popping out. So we are seeing kind of usual suspects in this case, and it looks like it's doing what it should be. But I think much more interesting is that most of the things that you might think are strongly adapted in any one species are not strongly adapted in other species. So here's the 4 taxa, so Annuus, Argophyllus, and then Pediolaris subspecies Phalanx and Pediolaris subspecies Pediolaris. And this is just this kind of index of how strongly associated a given gene is to in this case, this is Hargreaves evapotranspiration index. So this is the environment that looked like it was the most strongly repeatable. And you can see there's a few cases where there's, every single species looked like it had a response. We do see high repeatability in some places in the genome. Now, some of these things are probably due to drift. Some of them are false positives. But if you looked at the things that are most strongly associated in any one species, they are not associated at all in the other species. And so I think we'd be doing a disservice if we called this an example of high repeatability. There is certainly detectable repeatability above randomness, but most of what looks to be going on is non-repeated adaptation, or at least non-repeated signatures of association. Any questions about that before I go on? Because I'm probably covering a lot of ground here. Does that make sense so far? So Christian, that was Christian asking about the inversions. It's really hard to see who's who.
Yeah, I understood then
Okay.
how you get rid of it, with the assumption that the inversion is not shared between the species, yeah.
Yes, yeah. And so here, this is again a different approach. So now we're looking, in this case, this is the pairwise analysis. So not looking at one gene simultaneously across all species. We're looking here genome-wide between pairs of species. And we're saying, let's look at the window of repeated association and compare those to the non-windows of repeated association. So the windows that are a strong association in one species only, but they're not repeated in the other species. And so I think that controls for what you're suggesting here, because if there's if an inversion is driving a strong association in one species, it will show up into this block here. And so here we're looking at the proportion of non-WRAs that are in inversions or haploblocks versus the proportion of WRAs that are in haploblocks. And so we're seeing that if everything was falling along the one-to-one line, there'd be no enrichment of repeated evolution within inversions. But because we see many of the points between the pairwise comparisons between different species involving different phenotypes or soil or climate, many of our comparisons are enriched for these windows of repeated association within inversion regions. And so there's an inversion in one of the species, but the other species doesn't have the inversion, and we're seeing repeated adaptation, or repeated association at least. And we see that pattern in all, if we kind of use each species as a focal species and we look at all the other corresponding species, we see in most of the cases that fall significantly above or below the one-to-one line. The gray points are the cases that are non-significant by a permutation test. The strong colors are cases that are significant by a permutation test. And you can see that in most cases, we're seeing this big enrichment above the one-to-one line, which suggests that inversions are also driving adaptation. Regions that harbor inversions are also driving adaptation in the species lacking an inversion. So it's
Sam,
kind of...
Sam, sorry again
Yes.
for nagging on the inversions. So your data about the inversion status, this is done by sequencing all your individuals, so you have long read data, or is this on a species, I don't know. Where do you get this information from?
They were genotyped using Hi-C. We use Hi-C to try and reconstruct whether there was an inversion segregating or not. And Hi-C is used to kind of say which parts of the genome are falling close to other parts. And if there's an inversion, you can see that in the Hi-C signal.
You have Hi-C data for all of the individuals?
Not all the individuals, no. I think we genotyped specific individuals that we thought had the inversion versus didn't.
So could be, if I'm playing devil's advocate, that what we see in other species, that inversions are particularly helping for basically integration and adaptation to the same environment. Could this lead to a wrong signal in your test? Because you find in the same areas, you have the inversion driving it in one species, is also migrating to the other species, and then you would find it there. And this leads to the parallel signal.
Yeah, I'm trying to remember why we thought that was not the case. And so Lauren's group was doing all the inversion genotyping, and so I was less closely associated with that. But I think they spent a long time kind of trying to sort through that. And I know they did a lot of like phylogenetics, I think, of those regions. I'd have to look back at the papers to give you a better answer on that, Christian. But I think they were fairly confident that that was not happening. And and I think with the exception of maybe two of these cases out of in the neighborhood of 30 inversions, there were only 2 that were slightly shared between species. But I share that concern. I think and that would be actually also an interesting finding. So I think what I was intrigued about with this is that it's not just showing us that an inversion is likely contributing to local adaptation, but that the same region without the inversion is got strong enough local adaptation driving signatures that we're still seeing repeated association within that region. And so I think that what this shows us, or what I take from this, is that we're seeing both significant repeatability above randomness, but abundant redundancy and abundant variation in how these species respond to their environments. Probably a lot of this is also different phenotypic responses to the environment or lack of response in some species. So, it's much more complicated than what I can really get into here.
Can I ask a question?
But I think, yes, please.
You haven't mentioned interspecific introgression or interspecific gene flow. Do you differentiate this in your analysis, the things that have evolved in one species and just migrate into the other one? Is this a case of parallel evolution for you?
We haven't differentiated that here. I can, at the very end of this show, I can show you some slides about that. We tried to look at whether or not, and that's a great question. So the big issue would be particularly if introgression was happening, say, at the north of these species ranges, but not at the south, then the northern regions might be sharing alleles that are purely neutral, and the southern regions wouldn't be. And that would cause a decline in both species at exactly the same gene, totally violating the assumption that we had of independence underlying all of these statistics. So introgression would really be problematic for this. We tried to get at that by asking whether or not we looked at, on a window-by-window basis across the genome, what proportion of SNPs are shared versus non-shared between species in a given window. And we would generate a statistic for how many more shared SNPs they had than expected at random. And we didn't find that statistic was markedly associated with any of the things we've been looking at here. So we didn't find that there was higher sharing of SNPs between species in windows that tended to overlap the windows of repeated association or the inversions or anything like that. That was as good as we did. I think there's better ways to do that, but yeah, that was how we tried to tackle it. That's absolutely, I think, a huge issue that we got only part of the way to resolving.
Sam, can you explain to me again? I'm slow. Why do you think you have the excess of adaptation in regions with inversions? That's
so because of this part here. So here we're seeing these are the if we kind of think that the non-repeated the non-WRAs, these are examples where there's a strong association in one species but not the other, and the WRAs are cases where there's a strong association in one species and also a strong association in the other. If there was not enrichment within inversions, we'd see this falling on the one-to-one line.
No, I understand how you concluded from the data, but what's the what was the theory behind this? Why do you expect that inversions show this enrichment?
Well, I think it's more. I think inversions are persisting where they manage to capture multiple loci that are strongly associated with local adaptation. And when there are cases that have loci that are strongly associated with local adaptation, they also tend to produce local adaptation in the species lacking the inversion. I think it's showing us that the inversion is not critical to maintaining local adaptation, but it invades and it does well. Right? So it's not like you can't diverge without the inversion, but the inversion solidifies that divergence and invades. And so I think that I would say it's consistent with theory that would say that, there's relatively strong selection relative to migration at several loci. An inversion captures those alleles and suppresses recombination and invades. The species lacking an inversion still has those genes contributing to adaptation to some extent, not always, but some extent has those genes having mutations that drive adaptation. And we still see a repeated signature of association in the species lacking the inversion. I think that's my mental model for why this might happen.
So the inversions are basically passengers rather than drivers of the adaptation. Yes,
I guess you could say that.
Contrary to some of the other models, why we see inversions being so important for adaptation. Then, yeah.
Well, I guess important for adaptation is they don't seem to be the only way that adaptation can occur, but I think their prevalence within the species here suggests that they certainly are beneficial. Otherwise, I don't think they'd stick around as much as they have been. So I guess it's either, yeah.
Yeah, but if it can be done without what your data suggest, yeah, then the importance of the inversions for adaptation are not as generalizable as they are typically considered.
Yeah, I think that's a reasonable way to think about it. Again, I feel like I could go back to this data. I mean, you could stare at this data for many different ways, and sure, we're finding a significant enrichment relative to randomness, but that certainly isn't meaning that every single region that has an inversion has also got a strong signature of repeated adaptation. So we're kind of in this middle ground, and I wish that I'd done a better job of quantifying the effect size, and so I could tell you, 12% of the inversions had repeated signatures. I actually don't have that number handy. That would be a good number to be able to tell you.
Anyway, thanks a lot.
So I'm going to rush through a little bit the last two parts here, which I think shouldn't be too bad because they're relatively well, not simple, but hopefully relatively easy to understand. So because what we're doing is really just extending the kind of work I've talked to at length here in sunflowers. So Jim Whiting was a postdoc in my lab looking at 25 species of plants that had either whole genome or exome capture data with at least 6 populations. And this is previously published in most cases from all sorts of places around the world with lots of conspicuous absences of a lot of the global south. And so we'd love to see better data sets eventually and more species represented. But here's a phylogenetic tree of the different plant species we were including in this analysis. So going back, we've got conifers, 330 million years divergent from our angiosperms and lots of different species. What we wanted to do was take all this data through a common bioinformatic pipeline so that we would have the same error profile for all the different species, and then used OrthoFinder to identify orthology between all of these species, and then ran through the same kinds of association genetic stuff that I was talking about before, where we would do allele frequency environment associations within each species, no population structure correction. We would rank the genes based on the strength of their evidence for association within any given species. And then we would use this pick-min method to say, is a given gene enriched for high-ranking signatures of association across many different species? And that would give us a p-value for a given orthogroup or a gene family being strongly associated with an environment. I'm breezing through the methods. Are there any questions before I show you some results? So what we would see is, we're looking here is genes are in rows here, and our species are in columns. And these are all the genes that are significant at an FDR of less than 0.5. So we're using a very, very loose stringency here. But you can see that cases where there's a strong association in at least one climate are in black or colors, colors indicating more climate associations, versus no association in gray. And some of these genes at the top are showing up as being strongly associated in many of the different species. And so here's just the different environmental variables and showing you how many significant orthologs we found in each of those environmental variables. So lots of repeated adaptation in detectable across many, many different plant species, but also no particular gene do we ever find driving adaptation in every single species we're looking at. In lots of cases, there's quite a subtle signature of repeatability here. If we run a permutation test to just say how many hits would we expect to see under a null model, this is just showing you our 108 orthogroups that we find here is significantly much more than you would expect by chance, doing this lots and lots of times.
So Sam, what do you permute? Because you have a correlation structure in the phylogeny, so I'm not sure how you could permute it and take that into account.
Yeah, so there's no, we don't actually see any phylogenetic signal to this. And so that's, I don't know if I have that here. No, I don't. I have that at the back maybe. But there's no phylogenetic signature underlying this. We're not seeing more repeatability between species that are closely related and less repeatability between species that are distant. We don't find any of that there. You can see that because these are approximately ordered by phylogeny on the x-axis here. And you can see that there really is not a lot of that, but we also analyzed that formally statistically and found no phylogenetic signal. The permutation test here was done by just doing a random permutation of ranks, and just if we randomly ranked just random variables, how many times would we pull out significant hits? And we did this because here we're looking across lots of climates. Right, so we were just saying if a gene is an FDR of less than 0.5 in any one of these multiple tests, it's going to show up. And these different environments are all also correlated with each other. So there's a lot of pseudo replication going on here that we weren't really sure how to try and disentangle. So this is just showing us, if we do the same approach but randomly draw our stuff, we still see much more than we'd expect to see by chance.
Can I ask quickly? I see that your average, I don't completely understand what an RAO is. But
oh, sorry.
Like
that's an orthogroup that's been repeatedly associated. I should have. Yeah, so that's a gene that has a gene family that has fallen below this FDR of 0.5 limit.
Isn't your average very high? I mean, if you randomize your data, you get 40 genes that are repeatedly targeted by selection. This is what your randomization tells, right? It seems very high.
Yeah, so It's across multiple tests. So we're testing it in precipitation cold quarter and then testing it again. So we're doing the test in each environment and taking the intersection across, or the union of all those. So if it's significant in any one of these environments, we're calling it significant, which is,
Okay.
yeah, then that's why we're doing this permutation test. Does that make sense? So what's nice about this is we see a lot of the usual suspects for people that look at plants and adaptation. Flowering time, PRR 7 and 3 are these circadian rhythm genes, Gigantea, a circadian rhythm gene, auxin signaling, Rube. So lots of genes that people that know plant stress tolerance would kind of say this is kind of a laundry list of abiotic stress genes. So that's nice to see. We also found 32 orthogroups out of this 108 that had minimal functional annotation. And maybe those are some of the FDR false positives, who knows? But I think they're interesting candidates. I think we've taken a lot of pains along all this to be really over conservative. So I'm hoping that there's interesting stuff in those ones as well. The reason we took an FDR of 0.5 is we wanted to be err on the side of having as many true positives as we could. And that was because we then wanted to turn and get back to testing all this theory I talked about at the beginning by asking what is the pleiotropy of the genes that we're looking at. And so we tried to infer pleiotropy of these genes based on characteristics of a gene co-expression network. So we used data from RNA-seq data from Arabidopsis and Medicago and reconstructed gene co-expression networks. So expressions, networks representing, does a given gene tend to be upregulated along with another gene, then it tends to be co-expressed. And you can build a network of co-expression and then measure things like the network betweenness, the closeness, the degree, and the strength, which we could say we could use these as indicators of pleiotropy. So, you know, if you've got a high network degree, that means you're connected with many other genes. That's plausibly a more pleiotropic gene than a gene sitting out at the very edge of the network with a very, very low network degree. So each of these measures could be associated with pleiotropy. And we then looked at, for each of these, the Arabidopsis, betweenness, closeness, degree, and strength, and the Medicago, and then also a measure of tissue expression breadth. We see that the genes that are more significantly repeated, so the top category here being the one of interest, these are the genes that are candidates for repeated adaptation, are all tending to have higher pleiotropy than you expect at random. So falling on the right-hand side of these graphs is indicating that they're more extreme than you expect by chance. And so these are candidates for local adaptation, all had high pleiotropy. And I'm going to just leave that hanging there and transition to the next part where we looked at the same data set, but looking for global adaptation. So this is another postdoc in my lab looking at selective sweeps in the same data set. He excluded a few of the species because the exome capture data or the fragmented nature of the genome assembly that we were using wouldn't allow the sweep detection program to work well. And he also added in a few new species that he identified as having good whole genome data that would work for this. So we ended up with 17 species that had whole genome data, mostly a subset of the previous 25 species we used for the last part, and then used exactly the same methods to call SNPs and to use Pikmin to identify repeated adaptation. But here we used Omega Plus instead of the genotype environment association. So we're using Omega Plus to search for selective sweeps as regions that have high LD on either side of a breakpoint. And so then we're ranking our genes within a species based on the strength of evidence for being for harboring a selective sweep, and then asking across multiple species, do we see the same gene being very, very highly ranked in multiple species?
Sam, just a comment. So Omega Plus picks up hard but not soft sweeps.
Yes. Yeah, we are definitely not capturing soft sweeps. And we're probably not capturing some hard sweeps too. So here's the results oriented in the opposite direction as previously. So here we've got the genes on the columns and the species in rows. And the red squares are going around genes that are falling in the top 10%, or the bottom 10% based on the strength of evidence. So if we rank the genes, the ones that have the 10% lowest p-values are in these red boxes here. And you can see these are the genes that were Pickman significant after an FDR correction of being less than 0.1. And so you can see, particularly, we're seeing strong signatures within the Helianthus clade and within the eucalypts here. We were concerned that some of that may be driven by artifacts, because we use the same reference genome for each of these clades. So we have one Helianthus reference genome and one eucalypt genome. And if it's the case that there's a genome assembly error where, say, two contigs from different parts of the genome are stuck together in that reference genome, that's going to cause a breakpoint with high LD on either side and low LD across the breakpoint, which is exactly what Omega Plus is looking for. And so that could cause propagation of errors across Helianthus and eucalyptus. And so we redid our Pickman results, but each time using all 9 combinations of one sunflower and one eucalypt. And we're able to retain 15 of these genes that look like they have significant repeated adaptation or repeated sweeps across multiple species. So we find lots of genes for stress, again, abiotic stress and biotic stress, genes for cell growth. I'm not going to spend much time dwelling on the annotations because I think it's less interesting. What I really want to finish with is just saying that what I was fascinated to see is that when we look at the same indexes of pleiotropy, so high pleiotropy and low pleiotropy, here's our genes that are repeatedly adapted by selective sweeps, and we're finding that they are much lower pleiotropy than expected. So we've used the same methods to call SNPs, the same methods to detect significance, and we've just plugged in signatures of local adaptation versus global adaptation, and we're finding exactly opposite patterns in terms of the enrichment for pleiotropy. So here we're seeing high pleiotropy for local adaptation and low pleiotropy for global adaptation, which is the punchline of the whole beginning of this talk. I started off saying that, surprisingly consistent with theory. I think it is surprisingly consistent with theory, and we can argue about whether that's actually true. But I think it's seeing that this goes along with the framing I set up with at the beginning. I was surprised to see that. Another interesting result that Gabriele looked at here, our genes with repeated sweeps were highly enriched for having a large number of duplications per orthogroup. And so this is the null distribution. If we're randomly choosing from the genome, we're seeing lots and lots of gene duplication. And we think that makes sense, given that gene duplication probably involves subfunctionalization and neofunctionalization, which would come along with repeated sweeps. And so maybe gene families that tend to have more gene duplication also tend to have more sweeps going on. The direction of causality could be reversed, though. So I'm going to stop there rather than speculating, because I've talked for a long time. There's all sorts of stuff I could tell you about what I think this means, but I'd be more interested to just answer questions and make sure that this makes sense. Sorry, that's kind of a non sequitur to end on, but.
Can I start with a question? So in your global analysis, you have 17 plant species, and how can you be sure that you exclude some population structure in that global sample? What was your strategy?
Yeah, we didn't attempt to exclude population structure at all. We just included all of the individuals in this and ran Omega Plus on it accordingly. When we look at FST within these genes, so these are the species here that were driving the signatures, and then ranking the genes that were, in this case, any of these genes that are in boxed over in red here, so the genes that are driving the signature that are in the top 10% ranked by their signature of selective sweeps, those genes mostly tended to be not remarkable in terms of their FST. So we were concerned, oh, maybe we're just detecting local adaptation in the form of a sweep that's gone through part of the range. But that doesn't seem to be the case. We're seeing most of the genes that are interesting and contributing to the signature of repeated sweeps are not showing particularly strong FSTs. So this is ranked FST. So if they were in this case, low values of this ranked FST would be corresponding to a high value of FST. So it's kind of the reverse of normal. I'm not sure if that answers your question, though. It's
Yeah. Thank you. So I have another question.
Yes. Please go ahead.
Yeah. You put together all the association with pretty different environmental variables, as far as I could understand. And you still consider this as parallel adaptation? I mean, it could be that a gene would be involved in adaptation to one. I don't know how your data looks like, but you would say that if a gene allows adaptation to temperature in one species and to photoperiod in another one, then that would be parallel adaptation?
Yeah, no. So each climate variable was analyzed separately. So when we looked at temperature, we only looked at repeated adaptation for temperature. And then we identified the top candidates that were repeatedly adapted for temperature, and we called those significant. Then we did the same thing for genes associated with precipitation. So a gene could not be strongly associated with temperature in one species and precipitation in another. That wouldn't count. It had to be strongly associated with temperature in all species.
Okay.
Or was that not what you meant?
Yeah, I misunderstood this at some point. You said that I misunderstood it. And you started also discussing about the redundancies, like different, adaptive traits would have different numbers of potential contributors. You left that aside, or I just don't see it now. Like I was wondering, because you said you're not interested in the annotation, but the go categories, for example, would be one way to sort of evaluate the size of a given trait and see whether you get more often adaptation in parallel adaptation in go categories that are more reduced, or I don't know.
Yeah, I think it's there's so much more we could do here. And I think I think that would be a really great way to follow up on this. We only scratched the surface. I mean, data like this is so hard to reduce to anything, but I think that's fully important. I think to tie it back to where I started, what I think this shows me is that we're kind of in this middle ground where, how much redundancy do we have? This worldview is the kind of simulation land where there's full redundancy and architecture of adaptation is quite transient. Where there's no redundancy, you get these highly clustered architectures. I think we're kind of in the middle where there's some components of a trait basis are fairly non-redundant, and there's only a few ways of doing particular things, and then maybe there's a highly redundant component that underlies much of the rest of adaptation. We're certainly seeing detectable repeatability, which suggests that there are some constraints. We're not in this worldview where everything is totally interchangeable and you're getting a totally different suite of genes contributing to adaptation every time. But we're also certainly not in a case where there's, we don't find a single gene that has a strong signature in every single species that we study. So I think, we've only started to scratch the surface on this, but I think this is a really exciting area to kind of to keep poking at this data.
Yeah, thank you.
So a comment about other data that might be out there. So there's huge data sets on maize adaptability in multiple things, right? because most is basically adjustment to photoperiod. So there's a huge literature there with a lot of information that might be fun to mine.
Yeah, that's a fantastic idea. We started to do this at the beginning. We started with only pure wild species, and so that was one of our inclusion criteria. We're now repeating the sweeps analysis on all sorts of domesticated crops. So I think we've now got 25 crop species, including maize. Of course. So we're starting off by looking at sweeps there, and then we'll probably also repeat the genotype environment association eventually. But that's being done by a collaborator of mine at University of Hawaii, Mikey, and one of his postdocs, Mikey Kantar. So I think there's, and I'd be fascinated to get collaborators to help expand this however we can. I'd love to look at this in animals, look at things like altitude adaptation in animals, or temperature adaptation, freshwater marine adaptation. I think there's many different ways we can kind of see how this holds up in other regions that I think really just scratching the surface at this point.
Sam, I have another question concerning the predominance of high pleiotropic genes with high pleiotropic effects for local adaptation. So your interpretation of this is that these are basically genes with larger effects. Is this the interpretation, or do you think
that's what I would say? Yeah, and I mean, I think there's two possible explanations. I think genes, if it's the case that genes that have more pleiotropy have larger effects, and that pleiotropy tends to be oriented along the same axis of divergence, then that certainly makes sense. And I think, yeah, I think that would be my explanation for why we're seeing an enrichment there. I don't think there's any other plausible reason for it, really. I can't think of anything that would cause.
It could be that they are basically acting synergistically, that you basically look at evidence for synergistic pleiotropy. And this is a very important force for local adaptation rather than for global adaptation.
Yeah, absolutely.
Locally acting variants have been pre-filtered to be basically acting synergistically, and therefore they are much stronger.
Yep, absolutely. Yep. That's I mean, the single trait, if you want to frame it that way, you can kind of think of the single trait models of local adaptation, the trait being all of the traits that contribute to that environment, and that you're just ignoring all of the underlying mutational correlations and things like that. And that would then be totally synergistic effects, where a given mutation has effects on all trait dimensions in the right way, which whether or not that's plausible.
Did you say earlier that alleles with a greater degree of pleiotropy tend to be at higher allele frequency?
I didn't, no.
Oh, okay.
I'm agnostic about allele frequency here.
Thank you.
Yeah.
Other questions?
Okay. So, thanks, Sam. Again.
Thank you all very much. Really nice to. I wish I was there in person, but nice to chat.